And hi everyone, I'm Sunny from Thompson Rivers University in Kamloops. So my research is about sending survey to BC public about their willingness to pay for abandoned mine reclamation. And you can see the poster on the left side at the back, the orange one, yeah. Okay. So the upcoming technical session, technical session 5 is leveraging liquid bioblends to revive soil ecosystems in mine reclamation. The speaker is Joe Tobias, and is a founder of the Root Shoot Soils and a specialist in living compost and regenerative soil management. Since 2015, she has worked alongside farmers across BC, Alberta, Saskatchewan, and Ontario, supporting their transition from input-heavy systems to biology-driven practices rooted in soil health. Her approach centers on compost, not as fertilizer, but as a biological tool to reweaken microbial life, spark nutrient cycling, and restore degraded landscapes. Let's welcome Joe Tobias.
Hi, everyone. Wow, it's bright up here. All right, I guess I don't really have to introduce myself, but I'd like to start off my presentation with a quote. We abuse land because we regard it as a commodity belonging to us. When we see land as a community to which we belong, we may begin to use it with love and respect. So thank you for having me here today. My name is Joe Tobias. I am a regenerative soil and living compost specialist. I have been, since 2015, collaborating with farmers, mostly in Western Canada, but have recently embarked on an international project with sugarcane growers out in Thailand and helping them transition to biological management. So you're probably wondering how working in agriculture or working with farmers that are focusing on biological management is related to mine reclamation. I promise I'll get there. And my work centers on helping land stewards develop a better understanding of soil. And in my efforts to regenerate degraded landscapes, I found that high quality compost piles, and it's not the type of compost that you would normally think of. These are well-managed compost piles, and the point of them is to grow the biology, provide the habitat in these compost piles, and you basically take your workforce and you apply them into the soil. I think I need a stool. Oh. Yeah, it's just my notes. All right, it's okay. So before diving into the core of my presentation, I'd like to pause for a moment and talk about some concepts, and hopefully it'll remind us about soil and what soil actually is, and tie them with liquid bioblends. So what is soil? It's a community of living organisms merged with the non-living components acting as a system. It's not just a medium that we put together. It takes time for soil to evolve. And plants need a community of those soil organisms, much like we do, and that community interacts with water, air, minerals, and organic matter. It's multifunctional. It is where processes converge. You have the biological processes, chemical processes, as well as physical processes. It's complex, but complexity isn't something foreign to us. We deal with it every day in our relationships and in our work. And soil is one of those complex relationships that plants depend on. So first, let's take a look at the rhizosphere. What is the rhizosphere? Rhizo meaning root, sphere meaning world. The term rhizosphere was first coined by German scientist Lawrence Hiltner back in 1904, and he was a pioneer in rhizosphere microbial ecology. It is the region of soil surrounding plant roots, and it's the region of soil that is directly influenced by the plant's roots. It's poorly understood, but it serves as an interface between plants, soil, and microbes. So what's our understanding of the rhizosphere today? How big is that region of influence? It depends. The size can vary based on the plant species, the age of the plant, environmental conditions, the morphology of roots. As you can imagine, grass roots are different than tree roots, so their zone of influence is going to be very dependent on those things. And if you imagine a plant associating itself with mycorrhizal fungi, that myceliated relationship is also going to have a vast surface of influence. But it has been shown that the rhizosphere can extend from 2 to 80 millimeters away from the root zone. It is a complex system, and the interactions that take place inside the zone is influenced by chemicals excreted by plant roots and the microbes inside this zone. The soil here supports a dense population of diverse microbial community. In the rhizosphere, plant-microbe interactions drive vital ecosystem processes. It includes things like organic matter production. So the plant's root cap, it actually shows a rapid turnover of short-lived cells, and those cells are constantly regenerating, contributing to the organic matter matrix. Those cells, depending on the plant species, could still also be viable after several days. When they've sloughed off the root cap, they could still secrete enzymes and proteins, and they basically engineer the root's microbial ecology. Nutrient cycling is another vital ecosystem process. An example would be legumes partnering with rhizobia bacteria, and that partnership influenced nitrogen cycling process, or a plant partnering with arbuscular mycorrhizal fungi, and that partnership influenced phosphorus cycling. Disease suppression, it's been found that arbuscular mycorrhizal fungi doesn't just supply plants with phosphorus, nutrients, or water, but they've also found that it actually increases the plant's tolerance to pathogen damage. And finally, soil formation. Microbial cells and debris stick to fine clay and fine silt particles. Glues made by bacteria, fungi, and root hairs, they adhere to organic matter, and they pull different soil particles together, contributing to the soil structure, allowing air and water to flow freely. It turns out that plant microbial partners do not just exist around the root system. In the 50s, scientists actually began exploring microorganisms on leaves of plants. And it was F.T. Last, a British pathologist, who noticed a connection between pink yeasts and powdery mildew on cereal leaves. So we're talking wheat, barley, oats. So he was able to isolate different fungal species on grass plants. So he drew an analogy with the rhizosphere and concluded that plants have a phyllosphere. So the term phyllosphere has since been modified to include the total above ground parts of the plant. We're talking about the flowers, the stem, the fruits, the leaves, the seeds. Since the 50s, there has been a growing interest in understanding the microbial habitats and interactions found on all parts of the plant. So it's not just about the root system anymore. It's not just about the rhizosphere anymore. Every part of that plant harbors a diverse array of microorganisms, and those organisms interact with the plant, affecting its health and functions. Mining operations such as the one many of you visited yesterday often leave behind severely degraded landscapes, conditions such as compaction, extreme pH, poor physical structure. All of these basically disrupt ecological processes, and they reduce the soil's function, making it harder for vegetation to return, adding to the challenges of reclamation. Conventional mine reclamation practices pays little attention to the complex relationships between plants, soil, and microorganisms for long-term ecosystem recovery. So how do we restore microbial life if the soil looks like the picture on your right to begin with? How can we support those vital ecosystem processes like nutrient cycling, decomposition, organic matter formation when the soil conditions are extremely poor? Because in a lot of cases, in these types of systems, you have the organisms like protozoas and nematodes, microarthropods, they're basically done. Their populations are gone. And they indirectly affect vital ecosystem processes that plants need. So one of the best tools that regenerative agriculture has actually been integrating into their system is to use high-quality biodiverse compost distributed in a liquid form. So the key word here, it's a tool. It's not the end-all be-all. Ideally, it's integrated with other strategies. But the idea here is to help restore microbial life. I call these liquids, liquid bioblends, specifically compost teas and compost extracts. So quick disclaimer, the quality of the compost extract or your liquid bioblends, compost teas and extracts, is determined by the quality of their base ingredient, which is your compost. The compost has to be managed well. It has to contain high diversity of different species of microbes, microfauna as well, because you can't grow what doesn't exist. Which leads me to quickly discuss biosolids, which is a common material used in reclamation. Numerous studies, including a recent US EPA survey, identified more than 700 unique chemicals in biosolids, including pharmaceuticals, personal care products, household chemicals, industrial chemicals such as PCBs, PFAS, forever chemicals, flame retardants, and micro and nanoplastics, all of which carry unknown consequences for ecosystem health. In addition, the treatment process of sewage sludge, it eliminates beneficial species, beneficial organisms like your protozoas and nematodes that are required for nutrient cycling and decomposition as well. So if biosolids are used, ideally, it can be integrated with high-quality compost distributed in a liquid form to restore essential microorganisms for long-term ecosystem recovery. So the graphic or the video you see on the right is the typical way in which you would create these bioblends. Farmers now they have the equipment and they have the tools to make their own inoculants. So my role in our projects or my projects with my clients is to teach them how to make their own compost, teach them how to make their own bioblends so that they can reduce the use of fertilizers, reduce the use of chemical inputs in general, pesticides specifically. And at the same time, that process actually naturally restores those processes that plants require. So what are some advantages of liquid blends? Immediate nutrient availability is one of them. The soluble nutrients in that liquid can be directly accessible to plants and soil organisms for immediate uptake. Microorganisms that depend on water films, your protozoas, your nematodes, can reach the rhizosphere directly. They can go down to the root system, which enhances the establishment of that community where we need them the most. We need them around the root system. It's like our gut, right? Our gut needs the right microbes, or we need a microbiome. So do plants. There's also flexibility in application. Liquid applications can be delivered through different irrigation systems. I know that water can be a limitation on mining operations, but that can be remedied, right? I don't see why we can't catch rainwater at certain times of the year and have some water infrastructure on site that can support these types of strategies. Cost effectiveness, sometimes transporting and spreading large volumes of compost can be logistically and economically challenging. In contrast, if you have a small volume of really high quality compost on site, managed by the community even, and you have the right spraying and brewing equipment on site, you don't have to truck loads of solid compost to the site. So application volume and frequency can be adjusted based on soil conditions, which typical agricultural rates, we normally start with 100 liters per acre at multiple times of the year. Obviously, we time our applications based on the weather. Spring and fall is the best time. We really try to make sure that we're timing it with moisture, with farmers anyways that I work with, because microbes rely on water, right? We don't want to desiccate them. A lot of them go into a dormant state, but that's okay. We really want to support the ones that require that need to travel down to the root system. And customizable recipes. So recipes can be tailored by adjusting your compost feedstock even. So the compost that you're making, you can adjust it based on the plant community that you're trying to promote. So say, for instance, if you're trying to promote more shrubs, more trees, obviously the feedstock that you're using in your compost would have a higher ratio of carbon materials than nitrogen, for instance. So I'm going to get into a couple of definitions here. Compost extract, what is the difference between an extract and a tea? An extract, you're basically extracting the organisms, soluble nutrients, and the organic compounds that they built as part of the decomposition process. So it's real-time biological makeup. Versus a compost tea, you're taking that extract and you're adding microbial supplements and you're brewing it for an extended period of time. So what I mean by microbial supplements are your things like liquid kelps, fish hydrolysates. I'm basically giving them a seafood buffet when I'm brewing these teas. So the differences between a tea versus an extract, with a tea, it takes about 18 to 24 hours to make. An extract, you can make it from 5 minutes to 1 hour, depending on the concentration preference. So you can really tailor your recipes based on the conditions of your soil. Compost teas are typically applied as a foliar application, although there are some exceptions where it can be applied to soil. Versus a compost extract, you want to apply it into the soil and reach the rhizosphere. With a tea, you are adding your microbial supplements in the beginning of the process, versus an extract, you're adding it at the end of the process, and you want to add it right before you spray your liquid. You're not growing microorganisms in an extract, versus in a tea, you're activating and growing dormant microorganisms that are present in your compost. So a lot of times when I'm making my compost teas and I'm checking my liquid under the microscope, there is an increase in the protozoa population, for instance, or even the bacterial population, and sometimes the fungal population, depending on the organic matter that you used in your compost or your microbial supplements. Compost teas are generally used to prevent disease-causing organisms to re-inoculate the phylosphere. Disease is a big thing in agriculture. For instance, in our vineyards, there's powdery mildew in our vineyards, but there are ways to prevent those organisms from proliferating and affecting your grapes. And so one of the ways to do that is to inoculate the leaves, inoculate the entire body of that plant, and encourage competition, inhibition, and cooperation with the plant, because the plant has its own immune system. It does have the ability to combat disease or pathogens. It can call for microbial help if it needs to, and the microbes can work with the plant as well. A compost extract, it's generally an effective means to get beneficial organisms into the rhizosphere. You really want to promote nutrient cycling processes, and this is where you get your protozoas and nematodes in the root system. A tea is really easy to screw up. If you don't have the right brewing equipment, you can grow anaerobic organisms, you can produce acids that are detrimental for plant health, versus a compost extract, it's nearly foolproof. You're not growing, you don't have that lengthy brewing process that can lead to potential harm to your plants. So in summary, the application of teas and extracts is a means to effectively replenish the plant and soil microbiome for optimum growth and health. You have the tea for the phylosphere, and you have the extract for the rhizosphere. Really the idea here, and this is what I tell my farmers often, is So it's just you at the farm, it's just two of you at the farm, maybe it's a married couple. When you have the microbes to back you up, you have trillions of species that are growing those plants for you. So you better make them happy. They are your workforce. They are much better at growing plants than we are. So here are a couple of observed outcomes, obviously smaller scale. So this is definitely a scalable solution. What am I going to talk? Oh yeah, right. So this is an urban context. So these are bioretention cells by the Green Infrastructure Department with the City of Vancouver. BRCs are meant to filter stormwater pollutants from roadways before the water makes its way into our watersheds. And the soil substrate of each of these sites are typically 70% or more sand. So you can imagine that growing medium, it doesn't retain water or nutrients. If the plants are planted in the spring, and you have a really dry spring and these sites are barely irrigated, those plants are gonna die. They're not gonna thrive. And so my role in these types of systems is to go in there and hopefully support the plant with these microbial, their microbial partners. Next, I have the 60-acre parcel in Creston, BC. Farmers of mine, Ian and Kathy Finlay, they are the owners of this farm. They acquired this land back in 2020. A year later, Ian contacted me to help him revive the soil so they can transform this barren and bare land that was basically intensively managed for many years. And he wanted to turn this into an orchard for cider production. It took a lot of commitment from the farmer for this transformation to happen, but basically, we started working together in 2021, but we didn't actually get out to spray around October of that year because we had the heat wave. We were waiting for the right time of the year and catch as much moisture as we can, because obviously all this work of building compost, having the right equipment, it's a waste of resources if we're spraying microbes and they just end up dying because there's not enough moisture for them to begin with. So I helped Ian build high quality compost. He incorporated cover crops. So cover crops, he basically incorporated 14 different species of plants, grasses, forbs, legumes, brassicas, to help with that organic matter production, as well as helping build soil structure. So what he did was he's creating the right habitat in his soil so that when he goes and sprays his microbes, they have a home, right? They're not just gonna die off in the soil. And then the photo on the left, this is a recent photo. It's a young orchard. He just planted his trees back in 2024. And the picture on the right, this was 2 years after we started spraying liquid bioblends. So if you can imagine that transition, I don't know where these puffballs came from. They just appeared in the soil, so the spores were there to begin with, and we somehow activated their germination. So Ian and Kathy walked out one morning, they harvested some, dehydrated some, and ate some for breakfast that day. Liquid bioblend applications, it has demonstrated its capacity to increase plant establishment and biomass, improve soil aggregation and hydrological function. This is another interesting one where I worked with a farmer out in their farm is called Stonecroft, and they're out in Black Creek, and they saw a change in the behavior of water on all of their fields. Enhance nutrient availability and cycling, as well as shift plant community composition toward non-pioneer species. So again, another case study that I didn't show you earlier, farmer of mine, he just all of a sudden, one season of spraying these liquid bioblends, he was able to shift his fields from a weedy species into early successional grass. And this was something that he didn't expect, nor did I, and that was a good observation and great news on his fields. So there are several challenges to adopting this practice, but today I'll just focus on one, which is the spray. Oh, sorry, my bad. My other videos are not working. Oh, my bad, sorry. I wish this was playing so you could see how this works. Spraying liquid bioblends, it means using equipment that can keep the biology alive from your compost all the way to your soil. Oh, they are playing. Okay, sweet. So specifically organisms like nematodes and arthropods. So your standard sprayers in agriculture, a lot of them are not conducive to spraying biology. So farmers have had to custom their equipment so that biology can actually survive when they're spraying them out into the soil. Because the last thing you need is you're growing all these microorganisms in your compost, and then they end up getting stuck or dying before they even get out of the nozzles. So when it comes to reclamation, our best results will come from restoring the relationships within soil. That means reintroducing microorganisms at the right stages, if possible. So through seed treatments, that's another thing that farmers are now starting to understand. Before they seed their cover crop, for instance, they would inoculate their seeds with beneficial microbes. Or soil drenches. So plants have the support they need to establish and survive. But biology also needs structure, so pairing liquid bioblends with compost, woody organic matter, these can create stable habitats where roots and microbes can thrive together. We've already seen these tools improve water retention, nutrient cycling, and microbial diversity in agriculture and urban settings. I believe the next step is to test them in mine reclamation through pilot trials, co-designed with native vegetation protocols and shared learning across projects. If we take that collaborative approach, liquid bioblends have real potential to become a powerful addition to the reclamation toolbox. Plants and microbes have been doing this for a long time. I think we can really learn from their relationships and try and mimic what they're already doing. And with that, thank you for giving me the space and time to talk about what I'm passionate about. Thank you.
Thanks, Joe. That was great. We have time for a couple of questions.
Thank you for the presentation. I wanted to ask a question about one of the points you made on your slide, the use of the extracts on soils must be on soils that are greater than 5% soil organic matter?
Yes.
Is that correct?
Yeah.
So just thinking about reclaimed mine soils, you know, it depends on the site, but I would think that there are many reclaimed soils that are probably much less than 5%, so or soil organic matter. Do you think that the use of these extracts would still be somewhat effective in soils that have less than 5% soil organic matter?
Yeah, that's a really good question. So in the agriculture context, typically when I'm working with farmers, we tend to, I encourage them to submit soil chemistry tests to a lab and determine those values. And if we are seeing less than 5% organic matter, I encourage them not to plant their cash crops right away. I encourage them to build soil organic matter as one of the first things that they do in their management practice. So we take a phased approach. Maybe the first year, if you want to remediate, say, a piece of, when I'm working with my farmers anyway, if they want to remediate a section of their farmland, what we do is we focus on different species, different cover crops that they can incorporate as part of that process. So before they put in their cash crop, build organic matter first, and they can do that using different types of cover crop seeds. And then from there, after they've broadcasted their seeds, the seeds have germinated, we time it where we can then apply our liquid bioblends to those cover crops and get the cover crops as healthy and vigorous as possible. And after, when we feel like, all right, I think the soil is ready for your cash crop, then I would encourage them to mow down that cover crop. I would encourage them not to till. If possible, sometimes there are situations where we do need to till it into the soil, say we have really high compaction areas. But in this case, if they can mow it down and leave the root systems in place, because that's where the microbes live, and then they can use they usually use a different equipment to then drill the seeds of their cover crops, either the next year or the following year. And then the application of these liquid bioblends will be timed based on where the crops are at in their growth stage. So we really try to capture them, especially at the vegetative stage when when photosynthesis is that process is at the highest before say the tillering stage or something, if we're looking at grasses. I hope that answered your question.
Yeah, thank you very much.
Hi, Joe. Thank
Hi.
you for a great presentation. From a mining perspective, mining companies think about different things from a KPI that they're monitoring, that they can demonstrate that they're creating value. So besides the visual representation of these amazing photos that you've shown of regenerative landscapes, I'm curious if you're doing any work around the amount of greenhouse gases, specifically carbon dioxide, that regenerative soils could capture that could be helpful to demonstrate to a mining organization, like, by the way, I can help you with your greenhouse gas emissions.
Yeah, I don't know the answer to that question, but that's a really great, because a lot of farms are now about that as well. They want to capture as much carbon as they can in their soil. And carbon credits, all that conversation, I'm not really familiar with, but it's definitely an incentive to move forward with these types of strategies.
We have an online question.
Yeah.
Leah from Mining Impact Specialists. She says, great presentation. What are some of the limitations you could foresee in the mining industry regarding the liquid bioblends interacting with contaminants like arsenic, cyanide, high nitrates and nitrites, etc.?
Oh, wow, that's a loaded question. Does anyone have any answers to this? Liquid bioblends, I feel like it's not a silver bullet. There are other ways in which those heavy metals can be sequestered. We could employ or work with fungal organisms as well, and work with fungi as well as integrate these blends that have the bacteria, have the nematodes, have the protozoas, and almost allow them to not really figure it out, but somehow these organisms, they have the means to figure these things out better than we can. I know that's a very vague answer, but the limitation would be water on these mining operations. And I think this is a conversation I can take offline, and hopefully I can talk to that person more.
Thank you.
Sorry. Thank you.